Ladies and gentlemen, I remember to have been here about 20 years ago when my friend George Porter invited me to address the Royal Institution. And I remember all old friends like Peter Medawer, Francis Crick being here. But what I most vividly remember is that terrible clock up there. Because those lectures at the Royal Institution have to end exactly on the hour, and some bell is ringing and you have to stop. Now that was a royal institution. This institution is a more bourgeois institution and allows you only half an hour to talk. All right? Well, the subject, the foundations of life, were proposed to me by the organizers, and I think that was a very wise proposal. I would have been in much more trouble if the subject would have been named The Origin of Life. Well, the questions I will address are about the when, the how, and the how will include the question for frozen accident or necessity or law. And finally, can we recreate life in the laboratory? I could well start with some quotations of the lectures of this morning. The first speaker, Joseph Silk, said, "We don't know really how things began." That exactly applies to life too. The second speaker, Christopher Smith, said, "We do not need to understand the deep structure of matter in order to understand biology." I would agree completely with that too. And the third speaker, Carl Sagan, said, "To find anything like life on another planet would shake tremendously our views on how life originated on Earth." And he added, we then would be able to distinguish between necessity and contingency. Well, let me try to incorporate these questions into my lecture. Let me call the first part Life Itself, because that's the title of a book which Francis Crick published some time ago, and in which he forwarded the idea of a directed panspermia. Well, Francis did not send me his book, and when I asked him, he said, oh, I really hesitated to send you that book because I don't believe that, of course, myself. But I just wanted to provoke people to show them how little they know about the origin of life. And if you provoke crazy ideas, you can be sure that people will read your book. So the question is, what criteria do we really have? Well, if we go to the very early stages of life, we must say almost no criteria, because we have no witnesses. We have no fossils. We all know that even the simplest system of life as we know them must have been very complex. Well, at least the question which was asked in Francis' book was, when did life start? He thought it might be too complex, that it could have started within the 4 billion years of existence of our planet, at least in a state in which origin of life is feasible. Well, in fact, we have some criteria and some experimental data to answer that question. And that is the genetic code itself. No, that's the wrong one. This one. You know, the genetic code must have been very close to the origin of life, at least to that stage of life where you started to accumulate information, where you started to evolve protein structures, and so on. So there is one molecule which plays a key role in the genetic code, and that is the transfernucleic acids. It's a pretty short molecule, which you see in this picture. Is that shown correctly? Yes. We have nowadays about 2,000 of such sequences, and there is a way to use these sequences to tell you something about the time when this genetic code was formed. In order to make this story short, let me briefly tell you the principle how you could proceed. Among these 2,000 sequences, you have about 20 to 30 sequences of tRNA for a given anticodon, so a given molecule, which had come about when or which had been adapted when the genetic code came about. And you can follow each of these molecules through a number of different phyla. You can follow them through bacteria, eubacteria, archaebacteria, mitochondria through eukaryotic cells. And you can construct trees as is shown in this part. On the other hand, among the 2,000 sequences, you find also about 20 to 30 organisms, like human, like a given plant, like coli bacterium, like Bacillus subtilis, for which you have almost a complete set of tRNAs, which is about 40 different adapter molecules. And these molecules must have been assigned also at the beginning of the genetic code, but they traveled through different organisms, as I said. And they should have a topology of divergence, which is shown on the right-hand side. Now, first of all, if you do that phylogenetic tree of a given tRNA, and you can do it for many other ones, you can very precisely define the divergence of these tRNAs, and you get for the different nodes, which have been indicated here, a whole set of, let's say, 20, 30 or more tRNAs. So you can now take this set at given different time regions and compare it with the divergence of the original set. Or let me show it in the other transparency. You can compare the two divergences at different nodes, which are smaller, and the divergency of the whole. And now you can calibrate your time scale in relative units. Once you know when your first branchings, let's say between archaea and eubacteria, were, you can say how much longer it went back to the origin of the code. And since evolution in early stages was faster due to much higher error rates, you can at least determine upper limits. And the surprising result was the age of the genetic code, if you go to the origin, is only 1/3 to 1/4 a longer time than the dating back of the first branches in the phylogenetic trees. And since we assume that those branches occurred around 3 billion years ago, or a little earlier, or a little later, we can say that the genetic code can't possibly be older than, let's say, 4 billion years. And you find that on this slide. This is a relative dating which doesn't tell you about any mechanism. It only tells you the divergence among these early events before branching is not much older than the divergence we find among the organisms. And that tells you the genetic code is almost as old as our planet, but by no means older than our planet. And now I don't care. I don't care whether it really originated on our planet. If it originated somewhere in the universe, it would face the same problems as if it had originated on Earth. And since the conditions on Earth were, I think, uniquely better than at any other known planet, I would conclude from that the origin of life dated, as we heard this morning already from Carl Sagan, around 4 billion years ago at our planet. Well, then the next question of a principal nature you would ask is, what came first? The old scholastic question, the hen or the egg? Nowadays phrased the protein or the nucleic acid. Well, all mechanisms involved in that question are of such complicated nature that it would have been much easier if really hen and egg in the first phases of evolution would have been assigned to the same class of molecules, and there seems to be some evidence nowadays in the picture of the so-called RNA world. In other words, that the first RNAs were not only carriers of information, but also carriers of function, at least of some basic catalytic function, which is of making and decomposing nucleic acids and proteins. And if this were the case, this does not yet save us from the question of hen or egg, because if one given class of molecule has two functions, the two functions have different prerequisites, and you must find which feedback loop allowed to form this interplay between information and function. Now, all the other questions are unsettled. The questions, have there been protein-like substances before the nucleic acids? Most probably, because they are simpler structures than the nucleic acids. So there could have been lots of unspecified catalysts on the Earth. It seems to be settled the question that the first nucleic acid was RNA preceding DNA by various reasons. But again, the question, was RNA the first replicating molecule? The answer is possibly not. Possibly there was a precursor of the RNA which had a non-chiral element, the sugar causing much problem, as the work of Leslie Orgel shows. So these are all unsettled questions, and I will not further go into it because my subject is the foundations of life, not how did life originate on our planet, but how can life originate. Now let me then come to the second part, that is the question between necessity and contingency. Well, one could phrase this question in the following way. What is the difference between any, however complex, mixture of chemicals undergoing all sorts of reactions and a living organism, which after all is also a very complicated mixture of chemical reactions going forth and back? And the answer we would give to this is one word. It is information. Everything in a living organism occurs under the control of genetic information, highly regulated, highly controlled, and this is what we associate finally with perhaps with a definition of life. But then our question would leave, how can information originate? Well, we attempted, of course, then to counsel information theory as was developed in the middle of this century by Claude Shannon. But we will quickly see that this theory will not provide any answer to the question of how information can originate. Well, I will perhaps give a pre-answer to it. And that answer has been given in this country more than 100 years ago by Charles Darwin. And I would generalize it to my question. Information comes about by natural selection. But that I have to explain, of course. Well, first of all, we have to have a frame to deal with information. If we define information, as in case of genetic information, as sequences of symbols made up of definite classes of symbols, then we need a space in which each possible information or each possible sequence of symbols has a clearly defined point. So it could be a cellular or a point space, but it must be a tremendously big space. To give you an example, if you take a gene which has approximately 1,000 positions. And we know that the nucleic acids make use of 4 symbols. Then you have possibly 4 to the power of 1,000 different sequences. 4 to the power of 1,000 is about 10 to the 600. If for comparison you take our universe, or more simply, a sphere with a radius of 10 billion light years, and subdivide it into cells of a volume of 1 cubic angstrom, that's a volume which just would accommodate 1 hydrogen atom, then the total number of such cells would come up to 10 to the 108, a number which is negligibly small as compared to the number of possible genes. This will tell you immediately that to find an optimal gene which encodes a certain function cannot come about by a simple trial and error process. The physicists had great trouble, and I know from Eugene Wigner and Paul Dirac, who came and say this is a puzzle which might suggest that in order to understand biology we need a different type of physics. Well, what will come out is that what is wrong with it is you must not use that type of statistics to calculate probabilities, because if you would have to dice for these sequences, then it would be correct, and then our universe would be too small and too young, 10 to the 17 seconds, in order to get you anywhere. So we have to deal with a space, and we have to build up a space other than our universe in which each of the possible informations finds a place. And that's the 2 conditions we have to fulfill. The space must represent each possible sequence by 1 and only 1 point, and we have to order the points according to their neighborhood relations. In other words, points which differ in 1 position, in 1 symbol, should have a distance 1. Point differing in k positions have a distance k. You will see that gives you a metric like the street metric of Manhattan. And the metric is called the Hamming metric because it was introduced by Richard Hamming for binary sequences in informatics. And here you see such a space. Well, I started with, since we have no imagination, is that better? Yes. Since we have no imagination for dimensions larger than 3, we do it in an iterative way. We start with 1 position and connect those positions which can be transformed into one another. Then we add the 2nd position, and you see all we do is we double our first diagram and connect again the corresponding positions. And we add the 3rd position, again we double our diagram, get the cube, connect the positions. And now we go to the 4th dimensions, which we cannot imagine, but we have learned the algorithm. If you go to a dimension from dimension k to k plus 1, you double your former diagram and connect the corresponding points. Here you get a hypercube of dimension 4. This is for binary sequences. If you want to apply this to nucleic acids, you have 4 symbols which belong to 2 classes of bases. So you do it in 2 binary steps. You first assign the base class, the purine R or the pyrimidine Y. You come again up with a hypercube of the dimension nu, if nu is the length of the sequence. And then in a 2nd step, you decide which of the 2 Ys or which of the 2 R's. That means each point in this hypercube has a subspace which itself is a hypercube of the same dimension. And so your dimensionality of the problem is 2 nu if the length of the sequences is nu. Now, what are the advantages of this space? Well, let me show you how this looks for the 4-dimensional space. You have your 256 points. And now imagine if your sequence was 1,000 positions long. I can tell you how the diagram would look like. I can tell you exactly. It would be entirely black. Because there are 3 properties of that space. The first property is you have an unlimited volume. The volume is the sum of all the points. You know, each point represents 1 sequences. For nucleic acid sequences length nu, you have 4 to the power of nu points. Now, the second property, despite this large volume, the largest distances in this, or largest direct distances, which is the sum of the edges between 2 points, remains small. If you take our universe, where arbitrary distances usually of the length of 10 billion light years, and transform it into a sequence space, all you need is a nucleic acid of 180 positions. That means that every point of the universe is in a surrounding of about 360 angstroms, not further. And that is an important property, because in evolution, if you have any migration in sequence space, and that's what evolution is, you never have to go very far. You just have to know around which corner it goes. And for that, you need gradients, as on Earth. If a water droplet falls on Earth, it will follow the gradient of gravity. And in the same way, we have to put up a landscape in this space, which tells us where it goes. Well, the third property, finally, is also a very important one, and the connectivity in this space. You see that each point is connected to, well, of sequence length nu, in this case, to 2 new other points. Or if you can make little jumps, let's say if you can make a jump length of k being something between 5 and 10, your number of points is the nu by q. What is this binomial coefficient? So that is almost the kth power of nu, if you take an approximation. That means if you have a finite jump length in this range, each point is connected to an almost infinite number of neighboring points, and there is you really don't get as easily into a trap as you would get on surface on Earth. So these are the main properties, and now we have to put in some landscape. Well, first of all, you will ask, what is Shannon's information in this space? Shannon's information in the space is not any individual point, but it's just a property of the whole space. It's the entropy. Well, many of you may know what entropy is. Others perhaps might be satisfied with this cartoon from the New Yorker. I don't know whether you can read it. No, you cannot, but now you might. And well, but entropy is exactly the entropy of physics, which you have in statistical mechanics. If all points in our space would have the same a priori probability, then it would be simply the logarithm of this volume. That's what entropy is. That's what in Shannon theory is called information. Now, if the points do not have equal a priori probability, if their population follows some probability distribution, then you have multiply each with its probability and sum of the p times ln p. And but in any case, you see that entropy or information in Shannon theory is a property of the whole and not of the particular information you want to. And now comes a very important point. In the theory of Shannon, you assume that you can distribute your messages into all points, like an equilibrium distribution whose entropy you can determine. If you ask the question, how does information originate, you have to do exactly the opposite way. You have to concentrate into one point or into one region. And for that, you need motion in the space, and you need a corresponding landscape. Well, you have these nice lights that allows me not to see your clock. How can you move in the space? Well, we know that in order to keep information, in order to come twice to the same point, you have to reproduce. And reproduce is an autocatalytic reaction, which in its simplest case means that the dn over dt must be proportional to the n. So if n is 0, the formation rate is 0. But if n is finite, you can reproduce, and I will not bring you this mathematics. The true equations then have non-diagonal terms for the mutations and so on, but that can all be translated. I want to show you only the principle. If you now ask for the relative appearance of a given sequence I, that is the quotient of ni over the sum of all possible competitors, you have to differentiate this quotient. And you see you get the coefficient for the given one minus the average of all. And that's what selection means. If the individual coefficient is larger than the average, you have a positive exponential that grows. If it is smaller than the average, you have a negative exponential, it decays. But by the very fact that those with a large AI grow and with a small AI decrease, you have a self-organization until your average is equal to the maximum coefficient, and that's selection. Now, in the true world, as I said, you have to consider all the non-diagonal terms. You have to have non-linearities. I will not go into detail, but that is great fun for the mathematicians, and they indeed do have fun with it. Now is a question, you see already you move in direction of large A coefficients. So it's like attaching to each point altitude and evolution and is a mountain trip, but remember in an n-dimensional landscape and possible fractional, which looks more like Ansa Borrego or anything, if you want to have an example on Earth. Mandelbrot gave a nice example. He asked the question, how can a raindrop falling on an island reach the ocean? Well, apparently if you have a one-dimensional island, you have to fill up all these basins. But if you go already to dimension 2, you have at the rim points which are lower than the average, and the water will always flow out through the lowest point. Now, if you go on to the third, fourth dimension and finally come to Hilbert space, then each droplet will reach the ocean. And very similar, we are in a very high dimensional space with evolution, and we found that it's very easy to escape tracks if you do the right thing. And the right thing, if you do now the whole dynamics, you will show that what takes place in such a system is a phase transition. What I have plotted here is a calculation, a model calculation done by Peter Schuster, a sequence of length 50, binary sequence, reproducing with a certain speed and having one master which reproduces faster than the rest. If the reproduction would be very unprecise or practically stochastically de novo, what you would end up with is an ideal gas. You would populate all possible points, and your information is distributed over the whole space like an ideal gas is distributed. Now, if you reproduce precisely and make no mistakes, then you have survival of the fittest. That's only the best reproducing will survive, all the others will not be present. Now, the truth is in between. If you have a finite error rate, there is a very defined error threshold. Even for that length 50, you see you get here changes of 15 orders of magnitude. For length 1,000, it changes over many hundred orders of magnitude, where the information condenses or evaporates. And that's where evolution can take place. And if you do these conditions, you can do evolution in the laboratory as we do for several years. When we first started with evolutionary experiments, we were asked by people, how can you do that? Nature needed hundreds and millions and billions of years, and you know, an experiment you can do in the laboratory must not exceed the lifetime of a PhD. So, so the answer is you have to choose the right conditions, and we now have machines in which we optimize single molecules. So in conclusion of these basic to which we could add many detail, but I will not do that, view of the time. We could say there are a number of rules which clearly show that we have a mechanism which is dictated by some laws. And the laws are of a Darwinian kind. But it's not the very simple-minded Darwinian structure of stochastic mutation and deterministic selection. Rather, what you do is you do all in very large populations, which means you take your system in very many parallel channels. And then it means you get near the error threshold very far extended clouds of mutants, which are clearly centered to a given information, but which try many possible alternatives. And once you have a change in the environmental conditions, then you will have already a mutant which is better adapted in your cloud, and it will grow up. And that is the big trouble we have now with AIDS viruses, to which I will come in a minute. But the fact that you populate a large distribution means that you get a sort of pre-selection. Within this population, you populate only those states which themselves can reproduce, which are already almost adapted. And now you make a change. One of them is now better adapted, and it will grow up. And you have something like you would call punctuated evolution. You really have phase transitions. You have a distribution here, and there's a change, and then that will melt away, and it will condense in another region of the sequence space. So that's already true for the single molecules. Well, and if you look at it, this transition point where the phase transition appears is a clearly defined melting point, and this is the proof that nature really works along these mechanisms. We have studied a large number of viruses and have found they really all operate at the error threshold. Now, that's the trouble with the AIDS virus. The AIDS virus produces an exceedingly large amount of neutral variants. The polio virus does not, has the same error rate, but for the polio we have immune protection. For the AIDS virus we do not, and probably we will not have very soon any. But you see here the theory predicts exactly that, and nature belongs to that. If you look for the mechanism of virus replication, then it's quite complicated. We have studied that too. That gives you these cycles because, as I said, you have this interplay between genotype and phenotype. The single virus particle which enters the cell is just a piece of information, and yes, it is. Thank you. It is. It first has to be translated by the host machinery, and then some of its translation products have to feed back on the replication of the virus, and that is this complicated cycle with hyperbolic growth rates, a very complicated system. I will not go into detail. We call that hypercycle, just to show you that this exists in nature. Now, I have entered already the last part of my, the last section of my lecture. Can we recreate life in the laboratory? Well, we cannot yet recreate any autonomous organism, but by controlling the conditions, we can do essential steps in the laboratory. For instance, my students do experiments where they adapt random RNA structures to certain resistances to enzymes, or to certain replication rates, to certain ribozyme functions. That all proceeds for lengths of 100 nucleotides, which have a power of 4 to the 100 possible sequences. This appears within hours, and for total viruses of lengths of several thousand nucleotides appears in 2 to 3 days. So you see, we have constructed automated machines to study those processes. I was going to show you some, but perhaps here you see the problems which are involved. The machine started first with ordinary flow reactors in which we tried to grow viruses. Then we went down to smaller and smaller solutions. We took little droplets and did serial transfer experiments. And then we did thousands of them in parallel. At the moment, we are heading to study 10 to the 8 samples in parallel in an experiment. And I will tell you briefly what type of problems are involved. We take as a volume element, the volume element you can illuminate with a laser beam, which by confocal optics is focused into a space element of about a 10th femtoliter. That means its linear dimensions are about 1 micron. That's 10 to the minus 4 centimeters. So on 1 square centimeter, you can have 10 to the 4 times 10 to the 4 such little volume elements. And that we screen with our laser beam. We need for each volume element about 10 to the minus 4 seconds. So in 10 to the 4 seconds, we can screen all these 10 to the 8 samples. And 10 to the 4 seconds is a tenth of a day. A day is always 100,000 seconds. So you see, within ordinary time. And the consequence of this is we have evolved a complete new type of biotechnology, which we call evolutionary biotechnology, and which is now performed already in automatically controlled machines. Now let me come to the conclusion, the foundations of life. The question, how does life originate, is largely identical with the question, how does information originate? How can one create information? And this is information which existed long before man came about, namely genetic information. And we know there are very defined physical laws according to which information originates. And these laws tend to build up more and more complex systems. So it's really the study of the physics of complexity into which biology casts. Now the next question is, can one generalize those concepts? Well, to go back to the universe, which we heard this morning. Well, we know that in the universe, you have told us that there were also growth phases, like in the inflation period. Now, in such a growth phase, the system is always very far out of equilibrium, and you have very similar dynamical conditions as in the cases I have mentioned. In other words, you get instabilities there, and what grows out is this complex question of the dynamics of the system. How fast, how are the cross-sections of all the processes involved? And the question of why we find particular charge, why we find particular masses, might be a question which has similar answers to the ones I have discussed. But the other end is even more interesting, and I hope that Terry Sanofsky will come back to it. That's how does information come about in our brains? First, we handle information within our language exactly the way genetic information is handled. We put up symbol sequences. When we think, we reproduce and we mutate, and it's a very similarity. But how is that preformed in our brains? Is it also a neural Darwinism, as Jerry Edelman calls it? Is it reproduction of certain firing patterns in the network of nerve cells? That's a question which is open. So there are many questions which remain open, and I hope at the next anniversary of Nature, whichever you choose, you happen to choose, most of these questions can be answered. Thank you very much.
Thank you very much indeed. We have